Thank you, Lucy, for the introduction, and thank you very much for the opportunity to speak today. Today I'm going to speak about orchid conservation in Asia. Asia has got more than 9,000 different species of orchids, and they're concentrated about 5,000 species in the Sunderschild region, which is a tropical rainforest region, as well as Papua New Guinea, which falls in the same vegetation type. The tropical monsoon forests in Indochina have about 3,000 species, and the subtropical monsoon forests in South China and northern Indochina about 1,500. And also tropical monsoon forests in India have the lowest diversity with around 1,300 species. So when we look at the different vegetation types, how they look like, one which stands out, of course, is tropical rainforest. It's characterized by very tall trees, up to 50, 60 and more meters, up to 100 meters actually in Borneo. And so the forest floor is very, very dark, and orchids are only occurring as a large-leaved terrestrial species on the forest floor. But the lower main trunks usually do not have too many orchids. But in the crowns of the trees, higher up, there are epiphytes, but it's not as diverse as other orchid habitats. But still a certain high number is found in tropical rainforest. Another habitat type is tropical monsoon rainforest, which is a bit different to the evergreen rainforest because it has a strong seasonality. Usually between 2 or 6 months, up to 6 months drought can be found in tropical monsoon forests, and this causes the trees to be generally a bit shorter and the amount of deciduous trees is increased. So actually more sunshine can reach lower parts of the forest canopy, and therefore more orchids are found also at ground level and at the trunks and also in the upper canopy region. But nonetheless, the tropical rainforest areas have more species. The subtropical monsoon forest in southern China, northern Indochina is very similar to the Indo-Chinese tropical forests, but temperatures in winter drop down in many areas down to 0 degrees, which reduces the orchid diversity significantly. But still, a lot of species are found in this area, and the structure of the forest is still very similar to tropical forests. And then within each ecoregion, we have actually different special habitats. And for example, in tropical areas, usually up to 1,500 meters and higher, we can find tropical montane rainforest. So tropical montane rainforest is characterized by high humidity, permanent cloud cover, and very often a lot of rainfall up to several thousands of millimeters. So that kind of very humid conditions cause extremely high orchid species diversity. So a lot of the orchid diversity reported from tropical rainforest areas is actually found in montane forest. So as I said before, the lowland forest is rather species poor compared to montane forest types. And tropical cloud forest, like the picture here on Gunung Mulo in Borneo, is particularly rich in species. So hundreds of different orchid species can be found in such a habitat type. Also the mountains of Papua New Guinea also have a lot of cloud forest vegetation types, and very charismatic and horticulture interesting species like Dendrobium cadbersonii are found in that kind of vegetation type. Also, a special habitat are forests on limestone. And limestone areas you can have in the evergreen tropical climate as well as in seasonal climates. In the evergreen climates, like here, for example, on Gunung Mulo in Borneo, you have limestone outcrops out of the rainforest. And that kind of habitat very often is rich in slipper orchids, for example, as we see here, Paphiopedilum sanderianum, which is a very rare species restricted to the Gunung Mulo area. In the tropical monsoon climate, the forests on limestone are drier in nature. So you can see from the photo that the trees are a lot deciduous, have more deciduous species. There is a lot of gnarled and small trees growing on the outcrops, and a lot of actually the limestone itself is exposed. So orchids usually grow in the shadow of the gnarled trees or on the tree trunks, but hardly directly in full sunshine on the limestone. Only at higher altitudes maybe they can be found in such habitat types. And then the last type, which is very interesting in limestone, are the subtropical monsoon limestone forests, which are restricted to northern Vietnam, northern Laos, and of course southern China. And that area is famous for a lot of endemism too, and especially the genus Paphiopedilum is very rich in these subtropical forests. And one has to remark that these forests occasionally can even get frost for a couple of days. So temperatures really drop down to zero almost every winter or every other winter. Another special orchid habitat are tropical and subtropical sandstone and granite outcrops. So on the photo here we see Bako National Park in Borneo, which is a tropical sandstone plateau. And the poor nutrient levels on that sandstone habitat cause the trees to gnarl, and the trees of shorter heights, and therefore more sunshine can reach the trunks and the forest floor, and orchids can be found growing directly on the sandstone as well as on lower tree branches, basically at eye height. And a lot of endemic species are found in this kind of habitat, along with other very interesting plants as carnivorous plants. So for example, that area in Borneo is very famous for a vast diversity of pitcher plants. In subtropical monsoon sandstone and granite areas, like on the photos here, the left one from Hong Kong, the right one in northern Laos, orchids grow on granite boulders and shale granite outcrops. And so this kind of habitat can have also low temperatures, like in the limestone areas. Temperatures can go towards 0 degrees. But still, a lot of orchids can be found in these kind of habitats. So now when we talk about orchid conservation, so what are the threats to orchids? So of course the most important threat is habitat loss and disturbance. As we can see on these photos here, I mean, clear cut of forest, obviously all the orchid habitat is gone and orchids literally disappear after that kind of disturbance. If there is selective logging, for example for harvesting trees for charcoal production, like the kiln on the upper right corner, some trees remain and orchids still persist, but the microclimate of the forest is severely disturbed, and therefore certain sensitive species which need an intact microclimate may disappear. Another important threat to orchids is the collection for medicinal trade in Asia. So in different regions, orchids are collected for that purpose. And as an example, in traditional Chinese medicine, over 350 different orchid species are used. Out of that, about 97 are actually endemic for China. And most commonly, for example, here in Hong Kong, SAR, you can see pharmacies in certain districts which sell dried dendrobium stems, which are a common traditional medicine. And the quantities used for that are immense. So they're sort of sold by the ton, or traded by the ton. And so what kind of orchid species are found usually in the trade? Yeah, as I said before, mostly dendrobium species, but actually there are a lot of other species too, like Nervilia in the upper right corner. Barbophyllum species are commonly traded, as well as tubers of terrestrial orchids such as orchids or dactylorhiza species. So what is a very difficult task from a conservation point of view is to distinguish actually wild collected and farm raised orchids in the trade, because after drying and processing it's hard to distinguish even the species, let alone to tell whether this was farmed or not. And very often we have done some DNA analysis on purchased samples from the trade, and we found that actually different species were mixed, and so high value species were mixed with low value species because they looked more or less similar when dried, and so people can't distinguish. And so when we see poor stem qualities, then we think that poor processing conditions are usually found for wild collected plants when they're dried sort of in open conditions rather than a controlled environment. And very often, as I said before, a lot of the rare and expensive species have been replaced with cheap and common ones. The indicators for farmed orchids usually just can be seen on the packaging. So if there are trade names, they usually, and the species name is correctly reflected, then maybe it's a trustworthy source for sustainably sourced orchids. The brand name, trade names, and usually properly processed stems of dendrobiums, for example, from a sustainable source. The stems are clean without bite marks and mold. Another threat to orchids is the collection for the horticultural trade. So a lot of orchids have showy, attractive flowers throughout Asia, and very often the plants are collected locally and sold at roadsides to local traders, but also to traders which collect in larger quantities and often order from local villages orchids by the ton. And then they are brought to trade hubs and sold at larger quantities, either overseas directly or to local nurseries which cultivate the plants and make them look like propagated, and then they're traded under cultivated plants, but actually they're still taken from the wild. So there's a lot of laundering in this way going on in the region, and a lot of the showy species have become really rare in the wild now. So now we come to conservation. So what can we actually do? After, as a follow-up from an orchid conservation conference we hosted in Hong Kong a couple of years ago, we published in the Botanical Journal of the Linnean Society a framework which sort of gives a guideline of how to conduct a comprehensive orchid conservation program. So first, it starts with really understanding the taxonomy, ecology, occurrence and threats of orchid species of a region. And usually the easiest way of assessing the threats are to do a conservation assessment following the IUCN standards and use this to red list the orchids. And once this is known, one can do a ranking and prioritize species of highest conservation concern, and programs could be tailor-made for species which are the closest to extinction. In that process, of course, some mainstreaming is necessary and raise a public awareness about it and empowerment of individuals and institutes. And it's important that the findings have to be assimilated into government and corporate policy. And before starting any kind of applied conservation program, it's important to engage all the stakeholders and develop a conservation strategy. So we have done a number of studies in this direction, and today I would like to give you an overview of two case studies we have done recently, one in Hong Kong and one in Hainan in China. And so both studies were following the framework all the way from the beginning, point 1 to point 9, ending with capacity building after a successful reintroduction of these species. We applied the conservation framework in two case studies. One in Hong Kong SAR with the very endangered species Barbophyllum bicolor, and the second study in Hainan in China with the endangered species Phalaenopsis pulcherrima. So in Hong Kong we have done a comprehensive survey of all the orchid habitats, and finally this led to a conservation assessment of all Hong Kong orchids, which were published by Stefan Gale in a book called The Wild Orchids of Hong Kong. So with this kind of preliminary assessment, we were able to shortlist the most endangered species in Hong Kong, and then we prioritized these species and came up with a shortlist. And one of the top ranking species on that shortlist was Barbophyllum bicolor, which is only found in 9 populations in Hong Kong. And we were wondering, why is that species so rare? And to put some insights into that question, we launched a comprehensive conservation program for that species. So what we did was we started to do a conservation status assessment. So in Hong Kong, the species was listed as endangered. It's only found in secondary forest fragments along streams, and there are only 9 remaining populations, indicating serious habitat loss in the past and probably collection for the trade. No recent threats could be determined, and also there was no real evidence for any recent recovery. Although we saw a few seedlings, but it's very difficult to actually judge how old they were. So we were asking the questions, which factors contribute to the ongoing decline and hamper recovery? So we investigated the pollination growth biology of the species, and we did a population genetic survey. The growth pattern in Bulbophyllum is very interesting because usually the rhizome always makes one new bulb per year and keeps on growing along tree trunks or rocks. Occasionally also a rhizome produces 2 new shoots, and very often after a couple of years the back bulbs die and then 2 rhizomes remain disconnected, which means when looking at it one could believe that these are 2 different individuals, but actually they are the same individual and are clones of each other. So this is already a factor which we were wondering how many individuals then are actually found in the individual populations. Also, when we looked into the phenology, we found out that usually within just 20 or more days the flowers develop on a bulb. But it happens that different populations have different flowering times. So it very much depends on temperature and rainfall, which could be very localized during the monsoon, and especially in spring. And so we found that, for example, lowland populations flowered much earlier than upland populations. So there is a problem if there's a long distance pollen flow that actually populations are not flowering at the same time, so they can't be easily cross-pollinated. And not all rhizomes in every population actually flowered every year. So only between 42 to 86% of the rhizomes flowered at any one site. So which means there's a reduced chance of cross-pollinations because of these 2 factors. And then further on, we found that the species is actually strictly outcrossing. This was confirmed by bagging experiments. And no natural fruit set was observed in any of the local populations during 2012 and 2018. We observed some blue bottle flies actually visiting the flowers. They sometimes removed the pollen, but actually we never saw any successful pollination during this observation period. So then we hoped our population genetic survey could give us some insights what's going on. So in total about 513 leaf samples throughout the distribution area were collected. We analyzed 12 polymorphic microsatellite markers, and the results were quite shocking. So it turned out that there were only 28 individuals in total, of which 6 were only found in ex situ cultivation, whereas only 22 genotypes were found in the wild. And when looking more closely how the genotypes were distributed, as an example, in Hong Kong, there was only 1 population which had more than 1 genotype. All the others, obviously, were more or less clones. And so this is related to the previously mentioned issue that back bulbs die back, and then different distinct individuals appear as being distinct, but actually genetically they're identical and are still clones. So based on these shocking results, we immediately realized that this species is critically endangered and needs immediate conservation action. So then we launched a large-scale ex situ propagation and research program. And in 2013-2014, pollinia from flowers from 6 different wild populations were transferred by hand onto stigmas of flowering in the Botanic garden. And the capsules were harvested at maturity before dehiscence and approximately 20 weeks after pollinations. So capsules contained a very large number of viable seeds, and it was very successful to propagate them asymbiotically. We had to do 2 subcultures, and a large number of seedlings could be deflasks after 1.5 years. And importantly, because of the cross-pollination, distinct genetic seedlings were produced. As a first trial, we started large-scale reintroduction within the 90-hectare Cave Machine Nature Reserve area. And also we planted in botanic garden areas outdoors that species to also raise awareness with visitors of the botanic gardens. So hundreds of plants were deflasked and hardened, and seedlings were established either in pots or mounted on slabs and cultivated for at least 3 years before we used them into reintroduction experiments, as well as for the reintroductions within the KFBC Nature Reserve. So within the 90-hectare area, more than 300 plants were reintroduced on rocks as well as on trees epiphytically. So after 2 years, the first plants flowered in the wild, and very great was to observe that both color types, yellow and red, turned up in the seedlings, and they were observed in the re-established populations. So currently we're in the midst of monitoring the different populations to assess whether there is any cross-pollination happening. But so far we haven't found any evidence for that, but we're going to do this for the next couple of years. Another case study we worked on was the case of Phalaenopsis pulcherrima. It's a very showy, attractive species which was collected for the horticultural trade throughout its distribution range and brought it almost to the brink of extinction. The attractive flowers, usually pink in color, were used a lot for crossbreeding Phalaenopsis species, and a lot of the pink hybrids one can buy are based on Phalaenopsis pulcherrima crosses, and so therefore a lot of the wild plants were used in horticulture for breeding. So only 35 populations remain globally, and in Hainan province in China, our target area, only 6 populations remain, with only around 750 individuals left. And in a number of sites, the species disappeared completely. So the main conservation goal was to enforce the existing populations in Hainan and re-establish populations on sites where the species was lost, based on a scientifically driven plant reintroduction program. In this case, we had to do a lot of stakeholder consultation, as pointed out in the framework, and we established memorandums of understandings with Bawangling National Nature Reserve Administration in China, as well as the Hainan Wildlife Conservation Bureau in China. And the MOU was signed in 2015, which defined the roles of each party. So the aims and objectives of the study were to estimate the historic and current occurrence of Phalaenopsis pulcherrima in Hainan, and to assess the level of threats in Hainan. And we found out that the species is considered vulnerable at the moment. And we tried to characterize the ecological niche in Hainan and inferred the habitat specificity. And to quantify the spatial genetic structure, degree of genetic variation, and level of gene flow within and between wild populations, a genetic marker-based study was started to inform the reintroduction program, and to derive inference on the process of seed dispersal and population establishment. And after that, we developed a propagation and cultivation program to establish broad genetically seedlings to be reintroduced in different sites. So all this information was used to develop a full-scale reintroduction program for at least 3 sites in Bawangling National Nature Reserve, with more than 500 seedlings per site. So again, the global distribution of Phalaenopsis pulcherrima, indicated in red, is most of Indochina, and only small populations, and 6 populations are found in Hainan, the red dots. This species is restricted to granite rocky outcrops, but can also occur in open secondary dry forests. Our population genetic study clearly showed that subpopulations of Phalaenopsis pulcherrima reveal large pollen flow distances. So actually the armadillo bees, which you can see here in the photos, managed to fly more than 350 meters from one population to the next, guaranteeing long distance dispersal of pollen. And a further interesting observation from the pollination ecological study was that the species has a food deceptive pollination system, so it relies on a abundance of co-blooming rewarding magnet plants. So with a similar flower color to guarantee a successful pollination. So this was important to know for the reintroduction, because without the presence of those other magnet plants which are rewarding, it would not be possible to have a successful reintroduction and successful pollination after the reintroduction. So with this kind of knowledge, we started to propagate the plants, and more than 50 naturally pollinated capsules from different populations were collected and micropropagated by a commercial nursery. And around 2,000 hardened seedlings were available for planting after around 2 years. Several of the reintroduction sites were chosen in Botanic National Nature Reserve based on the habitat and the availability of the magnet plants. And in many of these sites actually had plants in the past, but because of collection they disappeared. So the main goal was to re-establish those population in these sites. So the hardened seedlings were taken out of the pots, and then we fixed them with an eco-friendly glue on bare rock. This was done by Kadoorie Farm and Botanic Garden staff, but also we had a lot of help from the nature reserve. They also, their staff helped us planting out hundreds of Doritis or Phalaenopsis pulcherrima seedlings. All the planted seedlings were labeled and monitored over the next 2 years to assess survival and growth rates. So very important in any reintroduction project is to assess the success rate of the program by monitoring the plants over a given period of time to see how many plants actually survived. So the program was pretty successful. So after 2 years, more than 800 seedlings survived, and some seedlings actually had already been pollinated. And we saw seed pods, and we also saw a lot of flowering plants. And so our reintroduction program actually doubled the number of individuals in the wild in Hainan. So we consider this is a very successful reintroduction program. So to summarize, what did we learn from our 2 case studies related to reintroduction and conservation of extremely rare orchid species with small populations? So actually epiphytic and lithophytic orchids can readily be propagated from seed. Seedlings for reintroduction can be produced asymbiotically within 2 to 3 years depending on the species. So there is no need for complicated symbiotic cultures. Site selection is a crucial consideration to ensure the species habitat requirements are met. Survival rates depend on it. Attaching seedlings to bare rock surfaces using a non-toxic bond is a viable reintroduction method. The reintroduction success can be assessed by regular monitoring of plant conditions of individually labeled plants over at least a period of 24 months, but actually it would be better to over a longer period of time to assess population trends. We achieved fairly high survivorship rates for both lithophytic as well as epiphytic species. And the onsets of flowering and fruiting among the reintroduced plants within 2 years confirms that ecologically viable populations can be re-established, and that it actually makes sense to do comprehensive reintroduction programs to avoid that species which were suffering from habitat destruction or collection for trade go extinct. So I want to thank you very much for your attention, and I would like to acknowledge my colleagues, Dr. Stefan Gale, Dr. Chi Hong Lee, and Dr. Pankaj Kumar, and all KPC staff who contributed to these projects, as well as our partners, the Agriculture, Fisheries and Conservation Department of Hong Kong SAR in China, the Botanic National Nature Reserve Administration in China, the Hainan Wildlife Conservation Bureau in China, and collaborators in Hainan, Professor Dr. Song, Dr. Zhang, both from Hainan University, and Dr. Hu is currently working at Royal Botanic Gardens Kew. Thank you very much for your attention.